In this lesson, we're looking at the lymphatic system and then also the body's defenses. When we look at the lymphatic system, it is going to be composed of two different parts. We have the lymphatic vessels and then also the lymphoid tissues and organs. Now, if we talk about the lymphatic vessels first, this is going to make up a very elaborate network, and that network is responsible for picking up excess tissue fluid and returning that fluid back to the blood. In this image right here, you can see that we've got the arterial system and the venous system. So that is circulating the blood around the body, and the blood is responsible for delivering oxygen and other chemicals to the tissues. Now, when that happens, there is going to be tissue fluid, which we call lymph, and it is going to need to be returned to the blood. But the lymphatic vessels are going to be responsible for really filtering this fluid before it does go back to the blood. So it is going to be a one-way system. It flows only towards the heart. And then other things about this system is that it's a low-pressure, pumpless system, and it really relies entirely on pressure changes and then also the milking action of skeletal muscles. This is a closer look at the lymphatic vessels. So the lymphatic vessels are shown here in green. They are right there next to the capillary beds, which are being shown in this picture. And their job is going to be to pick up that excess tissue fluid, the lymph, and return it to the blood. So substances normally excluded from entering the blood capillaries are going to be able to enter the lymphatic capillaries. And this would include things like cellular debris, bacteria, and viruses. So it will be the job of the lymphatic capillaries to really filter those substances out. There are going to be two different ducts that are primarily responsible for returning lymph back to the venous system. So this is circled here in red. We have the entrance of the right lymphatic duct, and then we also have the thoracic duct. So again, these are responsible for returning lymph back to the venous system. The lymph nodes are going to be working right there with the lymphatic vessels, and their job is going to be to help protect the body by removing or filtering out foreign material from the lymph. Now, when we look at these structures, they are enlarged structures, and we can see several of them here in green. And so the lymph is going to be filtered through them, and they're removing those foreign materials, and they produce lymphocytes that function in the immune response. In looking at the body itself, you can see that there are lots of lymph nodes scattered throughout the body, but they do get concentrated in three main locations. We have the cervical nodes, there are the axillary nodes, and then there's also the inguinal nodes. Now, the lymph nodes themselves, they contain macrophages and then also lymphocytes. The macrophages, their job is going to be to engulf and destroy bacteria, viruses, and other foreign substances. So they're pretty much eating them or wrapping themselves around those foreign substances. That's what we mean by engulf. And the lymphocytes, these are going to respond to foreign substances that are in the lymph. So here's a look at a lymph node. In looking at this, you can see that we have a region which is going to be called the cortex. And in the cortex, we have T cells that are going to be circulating continuously. And the job of those T cells is really going to be surveillance. So they are looking for foreign material. We also have follicles here, and the follicles are going to contain B cells that generate plasma cells which release antibodies. So the two of these together are functioning inside of the lymph node to help it respond to any foreign substances, bacteria, viruses, things that might be present in the lymph. We have a lot of other lymphoid organs that are working together with the lymphatic system. One of those is going to be the spleen. The job of the spleen is to filter and cleanse the blood of bacteria, viruses, and then also debris. And as it does that, it is also responsible for destroying worn out red blood cells. Other things that the spleen is going to do is it does store platelets. It functions as a blood reservoir, and then it also produces lymphocytes. The thymus is going to be responsible for producing hormones that help program the lymphocytes, so they will need programming before they can really function. Other parts, the tonsils, these are responsible for trapping and removing substances from entering the throat. And then another one is going to be what we call Peyer's patches. These are responsible for capturing and destroying bacteria before they're able to penetrate the intestinal wall. So when we look at the immune system itself really has two different parts to it. We have what we call innate or non-specific defense mechanisms, and then we also have adaptive, which are very specific defense mechanisms. We'll talk first about the innate defense mechanisms, and with this we have kind of a first line of defense, and then we have a backup system or a second line of defense. So that's going to be the first thing that we're going to cover here. The innate body defenses, this is going to include mechanical barriers that cover our body surfaces, and then it's also going to include things like chemicals and cells that act to initially protect the body from invading pathogens. So surface barriers, surface membrane barriers, that's kind of at the number one, top of the list when we're talking about the mechanical barriers here. This is going to include the skin, and it also includes some mucous membranes. The way that this is going to function is that, first of all, it may produce some secretions. And these secretions that come from the skin and the mucous membranes, these have a number of different functions. First of all, there can be acidic secretions that will inhibit bacterial growth. This is what happens in the skin, urine, sebum, the vagina. All of those locations have some acidic secretions that help to limit any bacteria from growing there. Another thing that the secretions can do is they may contain something like hydrochloric acid and protein digesting proteins. This would be what we have in the stomach. There are specific proteins that are going to digest. There can also be lysozymes. Lysozymes are found in the saliva. They're also found in the lacrimal fluid of the eye. We have sticky mucus traps. The sticky mucus traps, these have digestive and respiratory passageways. And then besides producing secretions, we can also have structural modifications. Structural modifications would include things like mucus-coated hairs. So this is something that we have in the nasal cavity. So it is designed to trap particles and substances that might be entering. We can also have mucus-coated cilia, which is going to be something that we do find inside the respiratory tract. Besides these surface membrane barriers, there's also going to be some internal defenses that are part of the innate body defenses. This will include certain cells and then also some chemicals. Now, natural killer cells make up a big part of these internal defenses. These NK or natural killer cells, these are going to be aggressive lymphocytes, and these aggressive lymphocytes are going to lyse and kill cancer cells, virus-infected body cells, and then also some other targets. These are going to overall be less picky, and so since they're less picky, they can remove really a variety of targets, which is why we can say that they kill cancer cells, virus-infected cells, some other things as well. So they're really aggressive killer lymphocytes. These are going to actually attach to the target cell membrane, and then when they attach to that target cell membrane, they're going to release lytic chemicals. And lytic chemicals are chemicals that digest things. So when they do this, they will function to destroy that particular cell that they are attacking. The inflammatory response is also going to be a big part of the internal defenses. As part of the inflammatory response, this is something that's going to be triggered whenever body tissues are injured. And when those body tissues are injured, there's going to be certain indicators, and these indicators are going to include redness, heat, swelling, and then also pain, and that will help to just the body detect that there has been some type of injury to the body tissues. This picture right here does show this inflammatory response. You can see highlighted in red right here all of these indicators that there is some damage that has taken place. The fact that we are going to have some increased blood flow into that area is going to cause the redness, and it's going to cause some heat, which will increase the metabolic rate of those tissue cells. And then we also have some pain and swelling, and that pain and swelling is going to really possibly temporarily limit the joint movement in that area. And all of this together is going to help with that overall healing process. If we look at an example of the innate body defenses, this image right here shows us an injury where we have a nail or a tack that has penetrated the surface of the skin. and now we have some inflammation there. What this is going to do is, first of all, we're going to have neutrophils that are going to enter the blood from bone marrow, and they're going to roll along the vessel wall and make it to that particular injury site. Those neutrophils are then going to leave the blood vessel, and as they leave the blood vessel, they're going to be drawn towards that place of inflammation. There's also going to be monocytes. These monocytes are going to be mobilized to that area of inflammation, and on their way, they're going to turn into macrophages, and as macrophages, they're going to be able to dispose of any remaining cellular debris. So in this way, we are responding to that injury that just occurred in that location. As part of the innate body defenses, we also have phagocytes that are going to be working right there in conjunction with the surface membrane barriers and also the internal defenses. These phagocytes include macrophages and also neutrophils, and we did see them on the previous slide in the image participating in that response to inflammation. The phagocytes are shown here in purple, and the first thing that they're going to do is they're going to stick or adhere to the pathogen. Once they do that, they're going to engulf the pathogen and bring it inside of them in the form of some type of vesicle, and then they will fuse it with a lysosome. That lysosome is going to contain some digestive enzymes like hydrolases, and it will digest that pathogen. And then once it has digested the pathogen, it's going to release the digested materials to the outside. Antimicrobial proteins are also going to be involved in the innate body defenses. This includes complement proteins and then also interferons. If we talk about the complement proteins first, the complement proteins are going to be circulating in the blood in an active state, and then when they come across foreign cells, they're going to attach to those foreign cells. When several of them attach to that foreign cell, they form some type of complex that's going to be able to create pores in the surface of that foreign cell, and then that causes fluid to leak out of that pathogen or foreign cell, and that is very detrimental to the survival of that particular cell. If we talk about the interferons are going to interfere with the virus's ability to infect healthy cells. So one way that it does that is it can interfere with the way that that virus actually attaches to healthy cells, and if it can't attach, then we're not going to get infection. Fever is another part of the innate body defenses. Fever basically causes the liver, the spleen. They're going to collect iron and zinc, and as they collect iron and zinc, that's going to prevent bacterial multiplication. So by raising the temperature just a little bit, we limit the growth and reproduction of the bacterial cells. The adaptive body defenses are also involved in the response that we have to foreign substances or injury. Now, when we're talking about adaptive body defenses, these are going to be antigenic specific. That means that these are designed to recognize something in particular. It could be a particular pathogen, a specific foreign substance, but there is a specific detection of that, and then it's going to produce a systemic response. That systemic response means that this is not something that just takes place at the site of infection. Instead, this is going to be widespread around our body. We are responding to that particular pathogen. This is also going to involve some memory. This means that it recognizes and it mounts a stronger attack on previously encountered pathogens. So if we get one infection by a pathogen, we respond to that, and then if it infects us again later, we respond even more strongly. The antigens, these are going to be any substance that's capable of mobilizing our immune system and provoking an immune response. The adaptive body defenses, these require lymphocytes. The lymphocytes are B cells and T cells, and then it also requires antigen-presenting cells, which we call APCs. The lymphocytes, these are always produced in the red bone marrow, and these, once they're produced, they're going to develop into either B cells or T cells, and that will depend on where they become immunocompetent. So there is going to be some training involved in these. In this picture right here, we're seeing production of the immature lymphocytes, and this is going to take place in the red bone marrow. Now, those immature lymphocytes, they are going to need to achieve immunocompetence, and depending on whether they remain in the red bone marrow and do that, and whether they travel out and then do that, will distinguish between T cells and B cells. So the B cells, they are going to remain in the red bone marrow, and they're going to achieve their immunocompetence there. The T cells, they are going to leave the red bone marrow. They're going to travel to the thymus in the blood, and then once they're in the thymus, they will achieve their immunocompetence. Once this has happened, those immature lymphocytes, they're going to leave the red bone marrow. They're going to leave the thymus, and then they're going to encounter their specific antigen at some point. Once they encounter their specific antigen, they will become activated, and after that, they are going to circulate in the bloodstream. Each lymphocyte is going to be able to recognize only one antigen. So it's a very specific recognition that we're getting here. So one specific lymphocyte, one specific antigen is going to be recognized. The antigen presenting cells, they are also part of these adaptive defenses. These are called APCs, and their job is going to be to engulf antigens, digest them, present fragments of them. These are going to be dendritic cells, macrophages, and then also B lymphocytes. When antigens bind to the immature B lymphocytes, this is going to sensitize or activate them. So on the surface of these immature B lymphocytes, there's going to be antibodies, and these specifically recognize a specific antigen. When we get that recognition, this is going to cause proliferation of clones. So we're going to copy those activated B cells, and these copied activated B cells, they either stay as memory cells or they're going to produce plasma cells. So these plasma cells produce a lot of antibodies. Many years later, we might have a secondary response that takes place. This would be a response that occurs by challenge of that same pathogen. This pathogen would be recognized by the memory cells, which are already activated B cells. And once those activated B cells recognize the antigen again, this is going to stimulate a much more rapid response. We will have plasma cells produced again, and these are going to secrete antibody molecules. If we look at how this is going to take place, if you look at the concentration of the antibodies when we have the primary response, the first challenge, you can see that it takes a little bit of time and there's not anywhere near the same number of antibodies produced as we have with the secondary response. So with the secondary response, everything's already activated, it's ready to go. So as soon as there is a challenge, we get a rapid production of the antibodies. We can acquire immunity by two means. We can either have naturally acquired immunity or we can have artificially acquired immunity. The naturally acquired immunity is going to be when we are actually exposed to a pathogen or antigen. So this is infection or contact with that pathogen. The antibodies, they have to pass sometimes from the mother to the fetus, and that would be naturally acquired immunity. We can also have artificially acquired immunity. And if we're talking about artificially acquired immunity, this is where vaccines come into play. Vaccines could be dead or attenuated pathogens. We could be exposed to those. We could also get injection of immune serum, so gamma globulin. There are certainly some benefits to vaccines. One of these is that we do avoid most of the signs and symptoms of the disease that would typically occur during that primary response. So we're avoiding the primary response and we're really just kind of jumping into the secondary response. And so the weakened antigens with the vaccine, they're still able to stimulate antibody production and they do promote immunological memory. One difference in passively and actively acquiring immunity is that with passive immunity, the B cells are not challenged by the antigen, and since they're not challenged by the antigen, there's not going to be any immunological memory. If we talk a little bit about antibodies, antibodies are going to be soluble proteins. These soluble proteins are secreted by activated B cells, or they can be produced and secreted by the plasma cell offspring that are produced in response to an antigen. These antibodies, they are responsible for binding very specifically to a specific antigen, and this is really where we get the specific recognition between the lymphocytes and their particular antigen that they recognize. Here we have a look at the actual structure of the antibody. The antibody is made up of two different chains. We have the heavy chain and the light chain, and there's going to be some disulfide bridges between those chains that help to hold them together. And then there's also some antigen binding sites on there. The antibodies are all going to contain a variable region and then also a constant region. The variable region is the region that's going to specifically recognize and bind to the antigen. Here we have a look at some of the different classes of antibodies. We call them immunoglobulin classes. You can see that they do have different names to them. They have slightly different biological functions, and they are produced and secreted in different places. In this image here, we are seeing how the antibody and the antigen complex is going to function. So the antigen-antibody complex, one thing it can do is it can inactivate things. One of the ways that it can inactivate is by neutralization. When it does this, it pretty much coats the surface of the pathogen and covers up the dangerous parts of that pathogen. It can also cause agglutinization, which is kind of a clumping together. It can cause precipitation. So of soluble antigens, it can cause them to precipitate. And this is all of those are going to enhance phagocytosis. We can also have fixing and activating of the antibody-antigen complex. And if we get fixing and activating of the antigen-antibody complex, this is going to enhance inflammation responses, and it can also lead to lysis of those target pathogen cells. T cells require the antigens be presented to them. And so these T cells get presented with antigens by macrophages or other antigen-presenting cells. And we can see the T cell here in the middle called the helper T cell. And helper T cells, they have a couple of different functions that they perform. One of those is that they are managers of the overall immune system. They are going to circulate through the body, and as they circulate, they recruit other cells to fight invaders that they do come across. They're also going to be responsible for signaling for antibody production. In this picture here, we're seeing the cell-mediated immune response. In this response, we have a cytotoxic T cell, and this cytotoxic T cell is going to bind specifically with some foreign target cell. If you look at the zoomed in picture of that, once we have that binding between the cytotoxic T cell and the target cell, the cytotoxic T cell is going to produce some different enzymes and release these enzymes, perforin and granzyme, and these are going to be released, and they're going to result in digesting and degrading of the cellular contents of that target cell that they've attached to. Here's just a comparison of the humoral adaptive immune response. So that's the antibody-mediated response. And then we also have the cell-mediated adaptive response, which is the one that's involving that cytotoxic T cell. With both of these, we do get production of memory cells, and those memory cells are very important for that secondary response if we are attacked by that particular pathogen or foreign substance again in the future. That way we can mount a much stronger response the second time around. If we talk just a little bit about organ transplants and rejections, there are 4 types of grafts that can take place, and when we do have these organ transplants or grafts, there is always a threat of rejection because that's something that would be recognized as foreign inside of our bodies. We can have autografts. This is when the same person is both the donor and the recipient. This is the best case scenario to have an autograft because that's really minimizing any chance that you're going to have rejection of that particular substance. An isograft is going to be when you have the transplant donated by a genetically identical person. Now, this is going to be really difficult to obtain because for that to be the case, there has to be an identical twin that we can get that transplant from. So autografts and isografts, that's the best case scenario, not always going to be possible. Allografts is the next best case. An allograft is going to be when the donor is not a genetically identical person, so it is someone else, Of course, if they are a close relative, that is going to make the chances of it being accepted a little bit better. We can also have xenografts, and xenografts is going to be when the donor is a different animal species. So that would be taking something from maybe a pig or from a monkey, a primate, some other organism, and transplanting that into humans. Allografts are the most common because, again, we aren't always going to be able to get the sample from the same person. We aren't always going to have an identical twin. When we're talking about the allografts, the best match possible is really key here. So really we need to have an ABO match, so the blood type needs to match or other blood antigens need to match, and we also require at least a 75% match of cell membrane antigens. All of the grafts that we have, they're going to require immunosuppressive therapy after surgery, and the closer the match that we can do, the less immunosuppressive therapy that we're going to need. If we talk about disorders of immunity, there are several different disorders of immunity. We can have autoimmune diseases. With autoimmune diseases, auto means self. This is when the body basically cannot distinguish between self and foreign antigens. And so what this means is that the body starts to attack itself. Examples of this, you've got a long list written here, but it's not an exhaustive list by any means. Rheumatoid arthritis is an example. Multiple sclerosis is an example. Graves' disease. And really, autoimmune diseases affect about 5% of adults in North America. So they are somewhat common when we're talking about them. 5% would be 1 in every 20 individuals is going to have one of these. We can also have allergies. These are hypersensitivities. Allergies are going to be when we have abnormally vigorous immune responses, where the immune system is going to cause tissue damage as it's trying to fight off this substance that it's perceiving as a threat. So it's perceiving this allergen as a threat, and it mounts a particularly elaborate response against it. Another example of a disorder of immunity is going to be immunodeficiencies. Immunodeficiencies are when we have abnormal production or function of immune cells or complement. Some examples of those are severe combined immunodeficiency disease, which is SCID, and then also acquired immune deficiency syndrome, which is AIDS. If we finish up talking about just a couple key points of development of the lymphatic system, the lymphatic vessels and the main clusters of lymph nodes, these are going to develop very early during embryo development. So by the 5th week of development, it's very obvious that we can see these clusters and these lymphatic vessels. And then most of the lymphoid organs, they're going to develop after birth, and that's really shortly after birth.